There will be one. So today we begin with subcellular localization. These three terms, they can be used to, for instance, predict function, can be used to predict subcellular localization. Can anybody explain to me what the difference between these three is, or just one term? Who can grab a term and explain what that means? Predict function by homology. What does it mean? Is it too trivial? Michaela?
Yeah, by homology it just means that you compare and that you have a set of sequences, a set of proteins where you already know the subcellular localization, and then you have some unknown sequences that you compare with the existing ones, and then depending on how homolog they are, you can infer some subcellular localization.
Mm-hmm. Yeah, exactly. So reading of the sequence similarity, you can infer annotations or copy annotations from another protein for which you have an experimental annotation if the sequence similarity is enough. And the word homology points to the fact that this is not a biophysical feature. It's not because the sequences are similar, they cannot fold differently or anything like that. But the idea is that there is an evolutionary constraint. The evolutionary constraints comes from when you go into a different organism, you still have to maintain the same some of the same function, and that is the constraint, to maintain function. And that is why from the sequence similarity you can actually see the evolutionary connection, and that evolutionary connection helps you to make this argument, this annotation transfer. The second word here is de novo. Can somebody please explain that one? The earlier you explain, the easier it gets. I don't know. Can anybody go for ab initio? Two terms left. Let's mix up. Yeah. Okay.
I think ab initio is methods are based on information which we can derive from the sequence, or
information
features, binding energy, or like.
First principle. So this is totally right. Binding energy is a perfect example. So where we in fact have some physical descriptor, such as a binding energy, something that we believe is valid for a protein and for a stone, or something like that. The Newton equations that describe essentially the movement between planets, or quantum mechanics, or E equals mc squared, or something like that. Those are generic laws that you typically derive Einstein's E equals mc squared from thought experiments, so without doing any experiment essentially. So C is the constant in this equation, and the constant speed of light, and that is something that you have to derive from experiment, the value, but not the relation. And so this is essentially what we refer to as ab initio. Now de novo.
You try to predict it without any additional information, or just from the sequence.
Yes, so that is true what you say, Daniel. Let me rephrase. Let me say it in a different way. So if you, for instance, have a machine learning device, the machine learning device does learn from information that is available for experimental proteins. But if you predict for a new protein, you do not need a homolog of that new protein in your data set. What you hope in this machine learning device, and that in some sense, Kyla, so from learning from features, you hope that you learn some principles. They are not ab initio equations. They are features. So charged residues have to do with binding DNA because DNA is negatively charged. That is not a law that you write. I mean, you can write that as an equation, but then it would be an empirical equation. It would not be a sort of an equation that you derive by Gedanken experiment, so to speak. While the de novo ones are those that are sort of extracted from a database. Okay? And again, the difference here is this is completely removing evolution. You try to now learn something that is specific to the sequence. But any sequence in the future, you assume will sort of follow that because of its biophysical principles. Okay? The problem, as you all know, eukaryotic cells or prokaryotic cells don't have a nucleus. Eukaryotic cells have a nucleus. About words, why is that obvious? Does anybody know what prokaryotic means? Or eukaryotic? What karyot means? Karyot is the Greek word. Is it written? You're smiling at me. Did I say that before, Vanessa? Or is it somehow... the way you smiled, I was afraid again it was on my slide. The Greek word somewhere. So karyot is the Greek word for the core. We typically refer to this in Latin or English as nucleus, in German too. So the one that has the nucleus, and pro is the generation of organisms that existed before the nucleus was invented. Eukaryote. So one thing is we have subcellular compartments, one thing is we have size. Why is it an advantage for a cell to have subcellular compartments? It's like little cells inside a cell. Why is that good? Yeah?
You have the possibility to have different concentrations and different conditions within these compartments.
So you have different pH values, for instance. Is that what you meant by conditions?
Yeah, for example, you also have higher concentrations of certain molecules. And you have different reactive environments.
Yes, that's essentially the pH value. So you have different environments. Yeah.
You can separate different reactions from each other that they don't interfere.
Which in fact is somehow also what leads to Michaela's density, increase in density, yes. Why would it help to have a different subcellular, different pH value? Why would it help to have a different environment?
If you want to degrade something, like in the lysosome, there's a high pH value.
Exactly, and this is sort of working together, right? So if you specialize your task to particular compartments, then you can make this entire process or the task that you do in that compartment work better in a particular environment. That's the idea, right? Anything else that you could think about? Yeah?
I would just say it's a lot more efficient as well if you separate the tasks by also.
It's not more efficient. Let's stay there. Yes, space.
Yeah.
So the efficiency has to do with both probability of hitting. So you want to make sure that a particular protein is next to another protein with which it interacts. In the nucleus you have 2,000 proteins and not 20,000, right? So the probability is just higher that you get the right one and that you interact with the right one. And since they are closer, you're also increasing the probability in terms of time. Okay, this is completely convincing, right? Now, I would argue that prokaryotes have undergone an amazing amount of evolution. So the kind of things prokaryotes do, if you look at the prokaryotic worlds, we may not like them because typically we encounter them. We only notice the existence of prokaryotes in us when we have a problem. But still, the evolution of prokaryotes is if you look, there's a podcast called Bacteriophiles. It's just outstanding what bacteria do. So why didn't they invent that?
I would actually counter that because as far as I know, prokaryotes also have some kind of subcellular compartments. I mean, they don't have...
Well, they don't have membrane enclosed ones, right?
They have...
So the first statement that you said about the environment, they don't.
I think they don't have like completely enclosed compartments, but they have something like mitochondria, like electricity.
They do have local densities, and they have essentially big machines, local protein machines that are aggregated. And if you like, the nuclear matrix is another example for such a machine. That does not nuclear matrix now doesn't happen in the prokaryotes, but it's a quote-unquote compartment that doesn't have a membrane around it, and it's essentially defined by a bunch of proteins that interact. And this is exactly what also exists in prokaryotes. It's true, but we still don't have a membrane enclosure, right? Your first statement was about environment.
But the other thing is they're a lot smaller, so maybe it's not necessary.
So maybe it's the size. And in fact, that is, I believe, that is a very crucial thing here. Yeah?
I think
Julian?
they're also more numerous, so they had... so you have to build a lot more of that stuff if you want to have... and that costs, of course, resources. And yeah, maybe...
Wait. So this is an interesting argument. So you say... so the cost of building the structure for the substructure or the sub-localization is so high that it's preventive if you want to have a high rate of
cells.
that actually, but so then you would expect that eukaryotes, well, that is true, but there must be a way to falsify the statement. The reproduction rate is so different between different types of eukaryotes, right? And in fact, some eukaryotes reproduce at the speeds the beat prokaryotes. So they're always prokaryotes that beat. They're always. So you're right. So I believe the top prokaryote is way faster than the top eukaryote. That's true. But there are prokaryotes that are slower, which is not a counter argument. I have to think this through.
I think that prokaryotes have this pressure to be fast, to get rid or to outcompete the environment. And in eukaryotes, especially in multi-cell organisms.
But do you believe that's what they started? So now you're talking about an evolutionary pressure. I'd argue that eukaryotes started as yeast or as little single cellular. Yeah, Michaela?
Yeah, I would say. I mean, I think recently somebody said that actually prokaryotes are not that extremely when it comes to evolution. I mean, if they have a very strong stress factor, for example, some kind of antibiotics or something, then they can quickly evolve. But when they don't, like when they're out in nature, people have observed that you don't have that much of a mutation.
No, that's not what I meant. So what I meant in terms of the remarkable diversity of prokaryotes is that they have adopted to all kinds of environments and in ways that are very often very remarkable. So I would, but anyway, so I believe we speculated a lot, and I believe we had some reasons, and maybe there are more reasons. Anybody else wants to make a point? Thank you. So this is a little bit more from the book of The Cell, the Alberts et al. Cell, a little bit more fine-grained distinction of different types of eukaryotic compartments. Let's get down to the simpler version. In the simplest version of it, you have somehow an extracellular space, you have the nucleus at least that defines the eukaryotes, and something inside the cell. That's sort of the minimal, and then the minimal comes with some other things such as at least the mitochondria and some other things. Okay, there is a pathway that you know about. So looking as a center from the cytosol, from the inside of the cell, there's direct exchange with the nucleus. That is a gated exchange. It's red because it's gated. This is sort of exchange in one direction. Essentially, proteins move from the cytosol into the peroxisomes, into the plastids, into the mitochondria, and that typically is the end. They stay there, and then there's this major path here called the secretory pathway, or referred to as the secretory pathway, where you go through the endoplasmic reticulum, then through the Golgi, sometimes to a variety of spaces, and ultimately you get secreted. Not all proteins that go into the secretory pathway here are secreted. Some are retained in the ER, the endoplasmic reticulum, in the Golgi, in other places. But this is yet another path. When we predict subcellular localization, sort of, again, the one way of doing it is through homology-based inference. This one we can do through sequence similarity or through text similarity. I'll get back to that. Then there are methods that use motifs, which essentially is sort of sequence similarity in a different sense, and for subcellular localization becomes a very different sense. And there are de novo prediction. There is no ab initio. There's no ab initio essentially in biology at all. The methods that use structure and the method that use sequence, and essentially we're going to focus mostly on sequence. Again, homology-based inference, you have a protein for which you have an experimental annotation, you have a protein for which you want to sort of predict the annotation, you have a high enough sequence similarity, so similarity between Q and E is higher than some threshold T, then you copy the annotation. That's homology-based inference. Now, what is high enough in T? Well, if they're very high, very, very, very similar, this will work. If they're very, very dissimilar, it will not work, and there's sort of a twilight zone in between. The twilight zone is sort of defined as the zone in which the signal disappears. The safe zone is when you essentially make this inference and you're always right. And the midnight zone is essentially defined as the zone when the signal is as good as random. So essentially using homology-based inference brings you something that is as good as random, right? That's the midnight zone. The work that I'm going to present comes from Rajesh Nair, here and up there, who is now at the FDA. So we begin with a data set of proteins for which we know the subcellular localization. We get a unique subset and run the unique subset against all. So we need the unique subset because otherwise we could essentially look at a set of proteins that are all similar to each other and we would have no information contained in that. We need to somehow find a unique subset. Okay, now what I want to establish here is the T for homology-based inference of subcellular localization. I say unique subset using the T. Sounds very circular, no? Is circular. How can I get out of such a vicious circle? So is anybody in the room not understanding what I mean by this vicious circle? So again, let's go back. I want to establish the T for the task of inferring that two proteins have similar subcellular localization. So what level of sequence similarity do I need so that I can say that I can in fact copy the annotation of the other one, right? E says a nuclear protein. At what level can I do that? Okay, and I say this is complicated because if I do this here, I use T to create this unique subset. Well, I cannot use T if I want to establish T. I need to do this. I need to start with something. What can I start with? Yes?
The subset of the known subcellular localization, and you can use that and you can hope that it's representative and then compute the similarities between these proteins and then compare how well the subcellular localization matches.
Absolutely right, Michaela. That's what I'm trying to do here, and that's what this image means. The question is, what T do I use to have this subset?
No, I mean the set of known subcellular localization is limited.
That is this. That's the blue. So green is all proteins, blue is all experimentally known subcellular localization. And that is too much over, so there's too much redundancy in there. So I need a non-redundant data set. So the simplest way to define too much redundancy, if I sort of look at all the ones that are 99% sequence identical, I already have 30% gone. So this is sort of a minimal way in which you could do it. But then arguably, or I can show you data that leaving it at 99% is also not good enough. We need to go further. What can we use? How can we sort of escape from a vicious circle here? Any idea? So some of you have been in the class last semester. So there was a T for let's call it 3D. So for inferring that two proteins have a similar structure, there was an HSP threshold, and there was an empirical value that told you when two proteins, when the similarity between two proteins is such that you can say that they have a similar structure. Right? Now, assume I took this T here to cluster. I took this T to say that is a unique subset because there's nothing in that unique subset that has a similarity above this T. Okay? What I'm trying to establish is T log. I'm trying to establish the threshold for subcellular localization. What relation do I have to postulate to escape the vicious circle? So if there is an equal sign between the two, then I'm just lucky. And what I did, I had a lucky guess, right? I started with a clustering that turned out to be afterwards right. I didn't know it before, and I did the right thing just with my first guess. what sign do I have to put between these two? What mathematical symbol? Yeah?
I don't really know. I mean, I think if you say the threshold of the 3D structure is larger, that means that you would be more liberal with that. And if you have more 3D structures and less 3D structures similarity, but more stringent.
So that's you said the important thing, Michaela. If I do it this way, then I'm conservative.
Yeah.
And if I don't want to sort of enter a vicious circle, I have to be conservative. What conservative means is my clustering is such that I'm not contradicting what I'm finding. If I do this, maybe I throw some of the data away. Maybe I make I get some problems because my data set gets too small. But as long as this is true in the end, my clustering was right on the T3D. Right? Is that clear for everybody? Because it means that my clustering is such that no pair in here has the same subcellular localization by that T. There's no pair in the unique for which I can apply homology-based inference for localization, since that one is larger. It is conservative in the sense that since it is larger, and if I had known the T lock before, I could have made this bigger. So then I would have had some more data, but it still would have led to the same T lock. In that sense, it's conservative. No? It's not clear? So there is some doubt in the room, is that right?
Yes, I'm confused.
Yeah?
Let's say, just for example, the T3D is, I don't know, 90%.
Let's put a number that is sort of closer to reality, is 20%. Okay? T3D is... Oh, yes. It's not only that you're confused. Thank you that you're confused. Thank you very much for being confused. Are you less confused now?
Yes.
That's very good.
So T3D is, I get this by comparing the 3D structures, or?
Yes. And it could be something else, right? As long as you have established the threshold for something that you believe is more conservative than what you're going to do. What you have to end, so the important point is your T lock will likely be different from the T3D, or from the T, let's call it to sort of make the story more stepwise, T first guess. Let's call this the first T first, the one that I use for clustering is my first guess T. And if the first guess T is more conservative means that the T lock that I find is in fact such that by this T lock I still don't have a pair of proteins in this set for which I could apply homology-based inference. So then for the T lock, this clearly is unique. Even something larger than it would be unique. It's not going?
What, by more conservative, you mean higher sequence identity?
So no, I demand them to be more distant from each other.
Ah, okay.
Conservative means I allow less overlap. So since that is, the clustering T is lower than the final T that I find, this clearly by my objective will end up to be a unique set. Is it clear? I take it it's clear. Anyway, so we have the set, and then we essentially look up in Swissprot. Now, the next issue that we have is the issue of, in my formula, of the similarity between Q and E being larger than T. In this, one issue is the similarity. So how do I actually measure similarity? One could be percentage sequence identity. So I simply measure the identity of letters. One could be the similarity according to the H-val. So the H-val is this curve here in blue, and I say that is empirically derived to distinguish between proteins for which I know they have similar structure and for those for which I don't. And then the H distance would essentially be, the H-val would be a distance from this curve, where plus values are above and minus values are below. And that's the HSP value. So again, it's shown here, plus 5, blah, 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 plus 25, and here minus 15. And then we essentially apply that. I thought I would start with a nicer curve in the beginning. What did I do? Yes?
So does that mean that we can look at the structure and the sequence similarly with respect to the localization? Because like what we discussed before was the...
So that is similarly, again, the word conservative means that the structure distinction is, let's call it the first guess. And as long as I verify that this statement here is true, that the first guess in fact is a conservative estimate of that T, I'm fine. Again, I may have thrown some data away that I could have used, but I clearly do not hurt myself by using what I want to find. And again, I'm sorry that I begin here with these confusing, slightly confusing curves, but.
My point is that the HSSP that we're looking at is based on the sequence, and the statement we made before was based on the structure. Can we like combine these two?
No, so that's essentially the HSSP is an empirical threshold for this blue curve, right? It is based on relating sequence to similarity in 3D structure.
Okay.
This is the curve that is optimized to, in fact, have a good distinction between the region where I can say that two proteins have a similar structure, and that is above that line, and those where I cannot say that. In that sense, this HSP curve relates sequence similarity to structural similarity. Okay? Not as a metric, but it does it in the sense that above the curve I have a very high probability of having similar pairs in structure. Okay, and again, it uses not only percentage sequence identity but also length, and it does it in an empirical. It does combine these two in an empirical way, which essentially the shape of the curve is not that surprising. So you can argue why the curve looks that way also through some theoretical assumptions of how you can change sequences if you imagine words. Okay, then we have these high values for HSP distance. So this is essentially curves that are very, very, very much up here. 0 is essentially exactly that curve. Minus 20 is this below this lowest red there, and the ones up here show you the precision. So meaning for each of these 5 different classes, cytoplasmic, nuclear, extracellular, mitochondrial, and chloroplastic proteins, if I put the threshold at an H value of 60, then all of them are above 95% accurate. The precision is of about 95% for all of those. If I go down to 40, well, it's still all above 90. And then you see it begins to drop. But sort of at the 0 line, so this is typically where structure inference works. For sub-cellular localization, it no longer does, because now for most of those, it's like 50%, which is not random, but clearly is not very high, right? So we're clearly here in, let's call it the drop zone. And if you want to have a sort of safe threshold, then you want to stay somewhere before this drop zone begins. So somewhere here, and this may be, the value actually may be 10 here, in that ballpark, okay? So in that sense, we verify that this statement here is true, that our clustering was right, and get sort of the first idea about the threshold. What else could we do to measure sequence similarity? Too trivial. Essentially use a significance score, an e-value, where you plot a score of the hit you find against the background of the database. You can do that in different ways, whatever the score is. For PsyBlast, it is Smith-Waterman score. You could have a different score here, but essentially you compute an e-value, and what I'm going to show you is in fact exactly the PsyBlast e-value, and then you sort of get a cutoff or a choice, whether you put the threshold somewhere where you have a high precision or somewhere where you have a high recall. And that ultimately is both captured by the confusion matrix, in which you show the number of positive predicted, the number of negative predicted. So you predict, for instance, that something, the 2 here, I assume you would predict nuclear against everything else. You predict something to be nuclear, you observe it to be nuclear, it's right. You predict it to be nuclear, you observe it not to be nuclear, it's wrong, and that's the confusion matrix, right? And that allows you to simply compute some of these ratios that are shown here. Again, I very often use the term accuracy and coverage as opposed to precision, and I got this one wrong again, precision and recall. And what is this one called? Anyway, so the reason mostly used are precision and recall for positives. Okay, I typically use accuracy for positives and coverage for positives, and accuracy for negatives and coverage for negatives if I have a 2-state prediction, because for me it's easier to remember that. It's 2 words, and it's a complete symmetry. If you look up on Wikipedia, then these definitions here have, whether you're in the positive or negative, we have so here it's called precision, here it's called recall, here it's called negative predictive value, thank you. Essentially, I want to prove my point here. No, it's not the same. It's exactly not this. That's exactly.
TTP.
Yes.
That's above. It's the recall.
No, that's not recall.
But that's exactly the same formula.
Exactly. Oh. you meant something else. Right. This is of course true negative.
Yeah.
But anyway, so it has a new name. So my point is here that the naming is asymmetric with respect to negatives and positives. And that is what I find very, very confusing. And I think you have to change that slide again. Okay, then there is an issue between PsyBlast and BLAST. PsyBlast does much better. Let's not go into that. But my point on this slide was supposed to be, this is another thing. This is the kind of thing that I've seen in master thesis present, so that you have percentage sequence identity, the e-value, the h-val, you have it for BLAST, you have it for PsyBlast, and you show all the curves. And now you want to say, is this better or that? Right? And from these curves, it's not easy, okay? Even if I gave you a lot of time, it is sort of true that this one here is better, this is what you expect, but it's not so easy to see. How would you plot it more directly? We cannot have the rock-like curve, because we need to find what threshold should we take. We're looking for the t. So in the rock-like curve is produced by dialing through the t. That's true. But we don't see the t. We don't see, in the curve itself, we don't see where is the t. And when we look at 2 different curves, or in this case that I showed you before, 6 different curves, then we cannot cut in the x-axis or the y-axis and just see for all the 6 the same value, because they are different. T's are completely moving through the curve. So what can we look at? Yeah?
suggests that you make two plots, one with a threshold on the x-axis versus the accuracy, and the other one with threshold versus...
Exactly. Something such as this. Where this is the h-val here, this is the e-val. Those are four different ways or three different ways of measuring t. So for green, structure, and blue is localization. That's what we're looking at. And you see that the blue is more conservative here. It's less conservative, so the green is more conservative than the blue. That's what we already said. So ultimately we verify the statement here. Now the challenge is, where do we put the threshold? And in some sense that depends on what you want to achieve with this, right? You clearly will not put the threshold here, that much is clear. You will put the threshold somewhere on this side here, but will you put it somewhere where sort of this is 100%, or will you put it somewhere where you sort of hit 90%? This is arbitrary and ultimately depends on how could you, what could you decide is the right threshold? You could ask your experimental collaborator and they have an expectation. That is one way of doing it. If they are not available. So typically homology-based inference is always the first step, and then you do de novo, right? So you could, for instance, look at when does de novo beat homology-based inference. So at some point de novo will be less accurate. Or at some point of this curve here, most likely you will not get de novo prediction of subcellular localization at 95. So everything above it, you will sort of assign by homology. If you can, right? You're with me on that statement? I turn around, if not. Let's just move on this one here. There is this one issue. You have now established the threshold T that allows you to do comparative modeling of subcellular localization, and you want to sort of publish this somehow, and the next question is, what else would you put into the paper, or what else should you look at in this paper? So you put a threshold, and again the threshold always has this business about where exactly do you put it. Do you put it a little bit higher? Let's say you put the threshold here. What else should you look at, the implication of that? How can you sort of measure the implication of that choice? So one is an implication that you see in terms of performance, right? But there's another implication that you could look at.
I would look at the false positives.
Yeah, but that brings us back to this curve, right? That ultimately is, what you're saying, Michaela, is you look at a different version of that curve. You look at a curve where you essentially again plot the t against some other measure for performance. That is in fact what you clearly should do. You're absolutely right. So I was sort of fast tracking too much here. If there is another way of looking at it that gives you a clear answer, then by all means that's the right way of doing it. So you should look at a couple of scores before you give in. But if for a couple of scores you always see something like that and you cannot quite distinguish, or even if you can distinguish and you say this is the score that I want to make, you still want to assess what it means in some other context. What is that context? What does it mean for the biology? So you can infer the threshold that I show here at 90-something percent, 95%. What does it mean? How can I estimate what it means? Yeah, my question. You're suggesting something, or I seem to ask questions today that are very confusing. I'm sorry for that. Okay, now you have your tool, you have your threshold. You're going to publish, but now you want to put something else into the publication. It's your name and all of this stuff, but some other data. What other data could you sort of look at?
Predict unknown proteins. And then
How would you do that? That's very right. What would you do as a proxy for unknown proteins?
we just determine the sequence similarity?
No. But I mean, what would you take? Would you take UniProt? If I were to be your supervisor, I would say, whoops, I don't have the CPU for that.
Maybe I take a representative subset.
How do you choose that? So do you cluster UniProt? Oops. This is getting difficult.
Maybe you can just limit yourself to one organism first.
That's the idea. So you're going to look at some sort of representative organisms. In this particular case, where there's an old piece of work, is yeast, fly, C. elegans, Arabidopsis, and human. And we simply, in this particular case, ask what fraction can we cover? So the real data is up here. This is sort of an increased view. Unknown is the white. Green is what SwissProt does. Blue is what this T does. And red is what I'm going to talk about next Tuesday. But you see, for instance, that the amount of blue here is very different between these organisms. And the amount of green is different. Explain to me why the amount of green is different.
I'd say it depends on which organism was more interesting.
Do you believe yeast is the most interesting organism? Don't you believe that? I mean, we are very biased people. Don't you believe that we feel we are more important than yeast? Well, some people like beer so much that they may not say that, but I would tend to argue that.
Maybe the easiest to study?
I believe this is a very, 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 very good one. So yeast is way easier to study. And is interesting enough for human biology because this is a eukaryote. And this is one of the easiest eukaryotes to do this. I believe this is exactly what you see there. I believe that Michaela is totally right. So interesting organisms are relevant. So you see priorities of interesting organisms. Human tends to be the most difficult one, yet there is a high prevalence here. Yeast is clearly the easiest one that is relevant here.